All right. So I have a lecture. This is going to be, clearly a recorded lecture. Um, that's just because I have to draw some stuff out or write some definition down. That's a little bit easier to do um, with a notepad than anything else. So this is where we're at for today. And so we're going to take some steps forward. We're still going to practice bioinformatics. We're still working with molecular biology. It's just we're going to change the data type and the questions that we can answer with it. So right up to this point, we've talked about phylogenetics, understanding how sequences change or how genes change, and how we can reconstruct evolutionary history and relationships and such. But now we're going to look at gene expression, right? We're gonna compare expressed genes. I'm still kind of debating exactly how we'll do that. Either way, I mean, what's going to happen is everybody's going to have their own tissue or sample that's going to be unique to you, that you're going to work with, and then we'll bring it all together and compare it all again, kind of in the same way we do with phylogenetics, but it's going to be a little bit different. But nonetheless. What we're doing is we're messing around with what we call RNA-seq data. And this is going to be what we would call the next generation or second generation sequencing. And so this is going to be Illumina data. And so Illumina really started, I don't know, picking up their pace in terms of their chemistry and how good it works and how often it's used, probably around 2008 to 2010, somewhere in there. I think Illumina starts to really pick up in popularity. And so we call this RNA-seq. Yeah, I mean, it basically stands for RNA sequencing. Unfortunately, we're still not quite in an area or in a time when we can do, end-to-end transcript sequencing, right? So again, we can't do that, but we can still work with these transcripts. So when we were doing phylogenetics, we had largely been working with regular DNA gene sequences, but now we're translating into RNA. Okay, and so any given tissue, I'll just go ahead and write this down, is any given tissue expresses about 10,000 genes. But remember, there are a whole lot of alternatively spliced transcripts present in the cytoplasm, things like that. So it's going to be a little bit higher, but it seems to be any time that I do an RNA-seq analysis, I've never seen more than like 12,000 genes present. So 10,000 seems like a pretty good number, and it's always about half of the genome. But every tissue is going to express a different 10,000 genes, right? Remember, I mean, this is still kind of what we're doing is practicing the central dogma. Right? DNA to RNA, to protein. Right? We're studying all of that stuff. Which segments of the genome are going to be expressed, what's going to be transcribed by, RNA polymerases. And we should recognize that whenever we're studying this stuff, we're messing with sequenced mature mRNAs. Mature. And so this means that we don't have any introns. None of our sequences will have introns in them. So RNA-seq, one of its big popular uses it helps us identify exon-intron boundaries. right? Because that's a little bit harder to make predictions about using math or any kind of algorithm or characteristics of intron exon boundaries. It gets a little tricky. But whenever we have the sequenced mRNA, right, it's a mature transcript. The introns are cut out. It's just going to be the exons in place plus the UTRs, right? That's going to tell us where exactly that mRNA came from, right? So it helps us place and identify both alternatively spliced transcripts and exon-intron boundaries. And so that's kind of the background of what we're going to be playing with. But in terms of how this gets generated, gets a little bit tricky. And whenever we start messing around with this data, you'll see that these files are going to be massive. Because ultimately how this is done is through kind of a shotgun sequencing approach. And what that means is that we're going to generate millions, if not millions, if not billions of RNA sequences. However, recognize whenever I say sequences, recognize this can, be translated into reads. So I may be saying read, I may say reads a lot, but really that's going to translate into sequences. I'm going to say these two things kind of interchangeably. So recognize whenever I say reads, I'm referring to unique sequences. And so Illumina will generate millions, if not billions, of reads that are all related to the expressed mRNA. But we don't know which individual read came from which gene. And that's kind of where the computers come in, right? That's where the technology leaves, and that's where the computers come in. Now, you kind of know me by this point. I'm largely a bioinformatician or computational biologist. I haven't done anything in the wet lab in a very long time. So doing this prep, I can't do, right? I'm not very good in the lab. And so prepping RNA or DNA for Illumina sequencing, I can't do. I can just process the output. But conceptually, what we're doing is so we have this free-floating mRNA, right? So that's going to be an mRNA molecule. And we'll see how good this actually goes. But not only do we have one transcript, we have thousands of transcripts floating around in the cytoplasm. Okay, so the first step that we do is extract the RNA, and then even further, isolate the mRNA. I don't know why mRNA, it feels red in my head, so it gets labeled red. But if we're not really familiar with like the molecular biology of RNA, I'm lifting this picture from somebody, but it's very clear, is that most RNA that's expressed is ribosomal, right? Because remember, endoplasmic reticulum are just full of, have a whole lot of ribosomes floating on them, and ribosomes are constructed from protein and RNA. It's just a specific species of RNA that's the ribosomal RNA. And so whenever you extract RNA and look at it under like gel electrophoresis, it's going to appear like this, right? This is going to be an image of RNA, like raw RNA extracted from a cell. Now I have done this step. I have worked with RNA up to this point, but once I get the RNA out of a cell, I give it to somebody else where they can do their wizardry that I can't perform. But if we're looking at this, right, we at least recognize electrophoresis, right? So anything that glows is going to be our molecule of interest. All right, we have a ladder over here that details or describes how big these molecules are, and the brighter the glow, the more of that molecule is present. And so whenever you extract DNA, what happens is you'll just see a single band, where you should see just a single band near the cell. It's going to be big, it's going to be heavy, it's going to be like 100 kilobase fragments. But when you isolate RNA, you'll have these two bands form. This really thick band for heavier weight molecules, and then a smaller, thinner band of the lower weight molecules. But both of these are ribosomal RNAs, right? And so both of these bands are referring to ribosomal RNA, both the 28S and the 18S. Gosh, I hope I'm not looking at my bacterial RNA. Hopefully I'm not. No, it's human. Okay, so yay, human. So this is what we'd expect to see. And so this 28S is just a fraction, it's just a part of the ribosomal RNA or the ribosome, and so is the 18S, right? It's just a smaller part of the ribosome. And clearly there's a lot of it. And so I sent out a video that kind of explains how we get just the mRNA out. Because if we just sequence raw RNA, all we're going to see is just all of this ribosomal stuff. And so we have to specifically isolate the mRNA that's going to be in these smudges. So if we get rid of these bright bands, what's left is going to be mostly the mRNA. Like the tRNAs will be down here, the small RNAs will be down here, but all throughout this smear is going to be the mRNA of interest. So we get rid of that. And we isolate the mRNA, because we always have to remember that mRNA can be differentiated from the ribosomal stuff because it's going to have all these poly A tails at the end of it, right? The polymerase will add a poly A tail to protect this mRNA from degradation as it enters the cytoplasm. I don't know if I mentioned this before, but RNA behaves a whole lot like wet toilet paper, right? If you just look at it wrong, it's going to fall apart on you and degrade and be totally useless. Really, really annoying thing. I swear it's just looking at it. I have certainly taken the same sample, extracted it twice, and one's going to look great and one's going to look like absolute garbage. And it was the same extraction. I don't know. It's tricky to play with. But anyways, we can isolate these mRNAs, or better people than I can isolate the mRNAs by taking advantage of these poly A tails. And basically grab these mRNAs that have a poly A tail and wash away everything else. And so what we do is we just sequence the mRNA. But again, mRNA isn't very stable. Quite honestly, I don't know how biology works because of this molecule, right? I mean, it's so pathetic. It falls apart so fast. It's amazing that we even work. So we have to convert it to DNA. Right, so part of this step, of this mRNA sequencing step, is to convert this into DNA with that's double-stranded, and it's through this process of reverse transcription. Like an enzyme will be present, like a reverse transcriptase will read the mRNA, right? It's single-stranded, and so it's going to build a complementary DNA strand from this mRNA, wash away the mRNA, and build the second DNA strand. So ultimately, what we actually sequence is going to be complementary DNA or cDNA. Whenever we reverse transcribe mRNA, we would call it cDNA or complementary DNA. And so this is double-stranded, right? 5', 3', blah, 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 blah. And so this we can work with, right? This is stable. We can freeze this and it will last forever. Right? This step has to be done before we can do any kind of sequencing. And so once we have our double-stranded DNA, this is going to get a little bit weird. I'll try to draw this out the best I can. We'll see how well this works. Let's say we have 5', 3', 5', 3'. And so we have this DNA that reflects mRNA. And so now we have to basically prep this DNA so we can sequence it. And so how that kind of works out is that we'll add these adapters to these sequences. And so these adapters, they're generic, they're made by Illumina. They're just going to be ligated to the ends of this DNA molecule. And then right, I'm skipping over some magic. And then we put this on an Illumina flow cell where this single fragment will be amplified, hundreds, millions of times. And, so like any good DNA technology, we have to sequence from 5' to 3'. And so ultimately what's going to happen is that whenever we do sequence out this mRNA or sequence this DNA, we're going to separate these two fragments. So we're going to make this single-stranded DNA, right? We're going to have single-stranded DNA. And so this adapter basically will initialize sequencing, right? So whenever we actually go through the whole sequencing process, again, I sent out some videos that describe this a little bit better. We're going to start sequencing from the 3' end inward in this kind of direction, because we can only add bases from the 5' to 3' direction. So we can only sequence, we're going to sequence first. If this is the forward orientation, we're going to take this read, or take this read and sequence it 5' to 3'. And so we'll get this fragment. And we get about 300 bases of it. Now, for most mRNA molecules, that's good enough, because mRNA is generally short. Most of the time, it's going to be shorter than 1,000 bases. Sometimes more, certainly, sometimes more, sometimes less, but on average, it's about 1,000 bases. And Illumina is to the point where we can get 300 bases out of a sequencing run. So we can sequence this strand inwards for about 300 bases before it quits. It stops. It's not as efficient. When Illumina started out, they were bragging about like 32 base pair reads. When they got to 100 base pair reads, that was a pretty big deal. But now they're about to 300 plus base pairs of sequencing. Now the data that we'll probably mess with is probably going to be between 100 and 150 base pairs. Anyways, so we're at least going to sequence this one fragment from it, right? So it will be kind of in this orientation, 5 to 3. And the amazing thing that with Illumina, so in the very beginning, when Illumina first came out, this is all that they could do. Is just generate one read from double-stranded DNA. And so we will call this like single end reads. It's just one read per sequence. Okay, but they got better at the technology. They kind of figured out they could do something called the paired end sequencing. And so paired end sequencing is, again, we're going to take this fragment of DNA, denature it so it's single-stranded, bind a primer that we can start sequencing from, and sequence in the other orientation. Again, this is going to be 5 to 3. And so we can sequence a fragment of DNA in both directions. And so this is called, and so we'll have ultimately another pair, another read. And so this is called paired end sequencing. Is one read from each opposite side of the strand. And they'll be oppositely oriented, and they'll be from opposite regions or opposite starting points of the sequence. Okay. And so ultimately, again, we won't know that these reads, or we can tell, hold on. I'm going to move this down for a second. Put it like that. And so the technology is to the point is that we at least know when two fragments came from the same segment of DNA, right? So we know that this segment and this segment are linked together. We may not necessarily know what's in the middle, but we know that these two things came from the same mRNA molecule. And so that's kind of a big deal, especially when we got to DNA sequencing, is that we can manipulate this insert size, right? And so this is going to be like an insert. Right, it's unknown, but these two things are linked, right? These two mRNAs or these two reads are linked, and this insert may be somewhere between 50 bases or 500 bases, whatever. We can manipulate this insert size. That's getting a little bit more tricky into DNA sequencing. I started it and I kind of want to abandon it because it's not very helpful for what we're doing. The main point really here is that these two reads are linked, and we know that they're linked together. They came from the same molecule. And that's the advantage of paired-end sequencing. So instead of getting one read that's 100 base pairs, we get two reads that's 100 each. And so it basically doubles how much of an RNA molecule we can sequence. And so nowadays, most Illumina mRNA RNASeq is paired end. On occasion, you can find old libraries. A library is just a collection of RNASeq reads. We can find multiple old libraries that are publicly available, but the more modern ones are generally going to be paired end and 150 to 300 bases. Very, very helpful. Now, sometimes these reads will overlap, right? Sometimes these reads will overlap. And sometimes we'll only sequence a fragment of RNA. So again, RNA has a habit of just breaking. RNA just has a habit of breaking. And so whenever we do an RNA-seq, a lot of times there's going to be many, many fragments of an mRNA molecule that came from the same gene, but it just broke, and so these fragments will be of varying sizes. So even if we have a 1,500 or a 3,000 base pair mRNA, we can ultimately sequence the whole thing because it's going to fragment during this whole extraction process, et cetera. This RNA will fragment, and so ultimately we can, say if we have, if I can kind of make this up. Right, we have a 3,000 base pair sequence that will ultimately be like fragmented, into overlapping regions. Sometimes they'll be longer, sometimes they'll be shorter, but ultimately these things will fragment. And we can sequence the whole gene, even with this paired in sequencing stuff, right? So all of these are DNA molecules, right? We can sequence that one in paired in fashion. We can sequence that in paired in fashion. I'm kind of going in the opposite direction, et cetera, et cetera, et cetera. And ultimately, we can take these reads and figure out what gene they came from. And the nice thing about RNA-seq and Illumina sequencing is that the number of reads per gene is equivalent to that gene expression. Right, so some genes won't be expressed at all, and we'll see 0 reads for that gene, right, because it's not expressed. Some genes will be more highly expressed than others, and we'll see more RNA-seq reads derived from that mRNA. And so ultimately, because of this, we can't measure gene expression for all genes in a tissue. And so this is kind of an advantage to say like our real-time or qPCR, right? qPCR is a lab technique that lets us measure the abundance of a single transcript, say among tissues or samples or something like that. But now we can do all genes at once. And all we're using is computational power to do so. Once we get the data digitalized, we can play with it. And so once we get to this, once this data is digitized, right? Again, we're dealing with paired in reads. And so what we'll have is whenever we're messing with this, we'll have a file that's going to be like file.r1.fastq. Right, this is going to be new. We haven't seen a fastq yet. We've seen a fasta, but a fastq is kind of similar, kind of not. And then we'll see another file that's called file.r2, come on, r2.fastq.gz. And so this file, you know, that's just insert your favorite word. Whenever we start messing around with public data, this will be like an SRR number that will relate to a library. But we're just going to call it file. What's important to note is this R1 and R2. And so with this R1 is basically going to be, these are going to be like the first reads or read one. And everything in this R2 are going to be the read twos. And so if I scroll up, that basically means this is going to be read one, and that's going to be read two. Right, so these individual sequencing reads will be put in different files. So everything that was initially read one will be in one file, and everything that was, once we wash everything away and redo the sequencing stuff, is all going to be in a second file that we call R2. It should be like that, R1 and R2. Sometimes it's just 0.1 and 0.2. Either way. And again, so these reads should be ordered. So if I kind of do this right, that will be like read 1A, read 1B. So I know that these reads came from the same RNA. Read 2A, read 2B. Right, so this will be ordered, and not necessarily in an ordered fashion from like smallest to largest or anything like that, but the pairs will be ordered. Right, the pairs will be kind of linked and kind of ordered all the way through. Now we'll see that these files are massive. These FASTQ files are quite large. We'll download, you know, 2 or 3 gigabytes worth of data, and all they are is just going to be these reads, all the way from read one all the way to, you know, read in. Right, all the way down. Right, and so there may be millions of reads in here, right, millions of them. On occasion, you will see, so nothing's perfect 100% of the time. This is definitely, biology is definitely, you know, not a super efficient process. And even whenever we go through sequencing, there will be some hiccups, right? So that you may see a read 3A that's not paired, right? So there won't be a read 3B. It's just some hiccup in the sequencing, right? Something is going to be a single end sequence, and not everything is going to have a pair, right? That's just, not everything is going to work right 100% of the time, right? So that will be skipped, and that has to be dealt with. All right, and so whenever we look at these FASTQs, right, we've seen the FASTAs, right? If we remind ourselves about FASTAs, it's going to be an open caret name and then a sequence. And so we will see something a little bit like that. But the fastqs are going to be quite different. And so a fastq format is going to be like, instead of starting with an open caret, it's going to start with an @ sign. And then we'll see the sequence name. And a lot of times it will be like -1 or -2, right? There's usually some number at the end to let me know this is the first sequence in a paired in reads. Okay, and then we'll see the sequence on the next line. And it is going to be DNA, right? So there will be T's, there won't be U's. We kind of have to make that translation in our heads, etc. Right? So we'll see a sequence. And below that, so in the older files, it would be the name repeated again, but now we'll see a plus line. And on the fourth line is going to be something totally different. They're going to be symbols. Maybe not @ but right, we're going to see special characters all the way through. And these special characters will be linked to each base, et cetera, et cetera, et cetera. Okay, and so basically each sequence, all the information of each sequence is going to be over 4 lines. And so what these numbers translate to, what these symbols translate to, is that these will be quality scores. Right? So again, nothing's perfect 100% of the time. And so we measure the quality of this base call. And so we measure this in terms of like a probability. And so these probabilities will be kind of on a log scale. It's kind of like saying so we'll see these backing up. So these symbols will ultimately be translated into a number. So this will be can be 10, 15. This is not actual, this is not literal. I don't know what each symbol translates to. But instead of writing down 10, 20, 15, 30, etc., it's just going to be a symbol is going to represent this quality score. And so we can make more symbols than letters. So between numbers, special characters, and letters, A through Z, we can basically cover the span of quality scores, because this quality, you won't see the quality be higher. I don't think I've ever seen a quality score higher than 50. I could be making that up, but I'll tell you what that means. I'm going to pretend like one's 50, but more often than not, I see 40. I think it may even max out at 40. I'm not entirely sure. I kind of forgotten. Okay, and so these qualities, again, it's just how likely this A that was called an A is actually an A. You know, how likely is this C actually a C and not some other base? All right, so that's what these quality scores are. And so these qualities, they'll range from like 10, 50, I'll do the whole numbers, 10, 20, 30, and 40. And so this is kind of like on a log scale. And so what this means, if we see a quality of 10, it means on average, if we see the score 1 out of 10 times called accurately. Okay, for a call of 20, this is saying 1 out of 100 bases with that score all called accurately. Um, 30 is going to be 1 out of 1,000, and 40 is 1 out of 10,000. And so the higher this gets, the better that quality is. And so remember, whenever we have billions of bases in this file, and if all of them had a quality score of 10, it means 10% of those bases are trash. Or sorry, it means, no, more than that. Yeah, okay. Oh gosh, now I'm lost. Everything's 10. 1 out of 10 is, yeah, it's trash data. Because it's basically every single base has a probability of 10% of being accurate. That's not good whenever we're talking about millions of bases. It almost means like we probably have to get rid of like 90% of our calls of trash. Right? That's not good. Even 1 out of 100, if we see a base core of a whole read and it's 100 bases long, it means at least one of those is miscalled wrong. Yeah, out of those 100 bases, one is for sure called wrong. That's the way to interpret it. 1 out of that base is called wrong. So yeah, I might have misspoke earlier, but it'd be like, if we had a sequence of 10 bases, one of those would be called wrong. And again, whenever we're talking about millions of reads, a lot of them are going to be called wrong, and we can't trust that data. So 1 out of 100 is better, right? If we see a sequences that are 100 base pairs long, all with this quality score of 20, 1 is wrong. It's not bad. That's okay, but 30 is definitely better, because it would say that for 1,000 bases that are all scored 30, only one of them is wrong. That's pretty accurate, and certainly 1 out of 10,000 is way more accurate, right? That's about as good as it gets. And so every single one of these bases will have a quality. And so that's what we put in a fastq. So it's going to be the name, the sequence, this plus sign, and then scores of how likely that base was called accurately. I should say kind of like called inaccurately. 1 out of 10 were called inaccurately. That's probably a little bit more accurate. I'm sorry if I confused anybody. But the short answer, to rephrase, the higher the quality score, the more likely that that's an accurate call. Okay, and again, so if we go back here, so this is going to be like fastq.r1, the fastq.r2 will be the same thing. It's just going to be as ugly at sign. Right, it will have the specific name -2 on it, right? So it will say that this sequence and that sequence came from the same RNA fragment. And again, it will be, a sequence, it will be a different sequence, a plus sign, and then quality scores that can be letters, numbers, special characters, etc., etc., etc. Right, so again, each sequence gets 4 lines. And so I think I want to leave it there. I'm going to call it there. And then on Wednesday, I'll go back to being live. And I'll kind of demonstrate where we can get this data from and how we can assess these qualities, right? How well we can assess, how likely these sequences are truly. Yeah, sorry about the hiccup and all of this. I feel partially embarrassed because that's gonna be public. Anyways, we'll pick up again with this on Wednesday.